Thanks. Great talk. So thanks, Martin. We're going to move straight on to a talk now on CRISPR approaches by Rachel Haurwitz, who's from Caribou Biosciences. So Rachel, over to you.
Good morning, everyone. So I'm looking forward to sharing with you a little bit of guidance and overview for methods for Cas9-mediated genome engineering. I'll be focusing specifically today on gene knockout strategies, though I'll highlight a few places where you can extrapolate to how to insert new genetic material as well. So I'll start with just a brief overview of what Cas9 genome editing is, though I'm guessing most of you are reasonably familiar already. I'll talk a little bit about methodologies for target identification and ultimately hopefully leave you with the understanding that calling genome editing is a bit of a misnomer, and that really it's all about DNA repair. So first off, I think probably everyone's aware that this system is based on a protein called Cas9. It's a DNA endonuclease that's able to introduce a double-stranded break in target DNA. Now, on its own, it's ineffective. It requires a guide RNA molecule to actually target it to the appropriate location in the genome. What I'm schematizing here is actually the dual RNA system. It's a CRISPR RNA plus a tracer RNA. The tracer RNA is really a structural module that helps Cas9 move into an active formation, whereas the CRISPR RNA is actually what does the targeting. And so what I'm showing here is that really the business end of the CRISPR RNA is 20 nucleotides long. And I should highlight that this is specific to the Cas9 from Streptococcus pyogenes. The general theme is the same for other Cas9s, though the specific length and some of the other specific sequences vary from organism to organism. In this particular case, the experimenter is able to simply dictate those first 20 nucleotides. It will base pair directly with the target DNA only if a particular motif called the PAM, or protospacer adjacent motif, is located immediately adjacent to the target site. So when all of this is true, Cas9 is able to engage with the target DNA, actually melt the duplex, form Watson-Crick base pairs between the CRISPR RNA and the target strand, and ultimately insert a double-strand break. Using this break, of course, is how we actually do the editing. And so this can lead on its own to gene deletion simply by the cell repairing the break. So it turns out cells don't really enjoy having their DNA broken, and so most have a variety of ways to quickly try to repair that break. And I'll talk a little bit about that in a moment. Additionally, though, of course, this is a way if the experimenter delivers donor DNA, which could be short single-stranded or double-stranded DNA, or long, say, vectors. This is a way to precisely insert new material at the broken site. So I'll highlight the workflow that we prefer at Caribou. There are a number of ways to actually go about achieving genome editing with Cas9. Our preference is to deliver these reagents directly as a ribonucleoprotein complex. So we'll purify bucket loads of Cas9, pre-complex them with either CRISPR tracer duplexes or single guide RNA, which is a fusion of those two, and directly deliver these into cells. We'll do that via nucleofection. And the reason that this is our favorite methodology is we, and many groups have now observed, this leads to the fastest kinetics of editing and the highest efficiency of editing. Additionally, it tends to result in the lowest off-target effects as well. I'll highlight off-target a little bit towards the end of the talk, but I will mention that the reason we think this is true is that by delivering an RNP, you get a quick transient burst of the RNP present in the cell where it can act on the particular target. If instead, for example, you're delivering via lentivirus or other mechanisms that might lead to high sustained expression of the molecules, you're only upping the chances that you will not only experience high on-target, but also high off-target editing. Typically, our workflow then results in about 48 hours later, lysing the cells and isolating genomic DNA, with the ultimate goal of being able to measure with high precision whether we were able to achieve the changes at the target site that we're interested in or not. So typically we do targeted amplicon sequencing and then deep sequence. So as we think about early stage target identification, it's important to think about the specific nature of the change that you want to achieve. What I'll be focusing on today are workflows relevant to attempting to knock out a particular target gene. So typically, you'll want to think about targeting an exon, and you'll typically want to be as close to the 5' end of the open reading frame as possible. Now, many of you may be aware of a variety of bioinformatic tools that are available to help you design guide RNAs. I would say, by and large, they're not much better than trying to aim at a dartboard while blindfolded. And one of the nice things about this system is it's very high throughput and easy to use. And so our recommendation is actually to simply empirically test each potential target site within your gene of interest and simply pick the winner. And through the rest of the talk, I'll help guide you by what I mean the winner should be and some of the key characteristics there. So for some example genes schematized here, we would simply look at every potential S. pyogenes Cas9 guide that could be designed that follows the rules of a 20 nucleotide target adjacent to a PAM. Now in this case, we're actually showing intronic guides as well. That may be relevant for certain knock-in approaches or various specific needs for a certain target, but in most cases, you'll be looking specifically at exonic genes. Now, what I recommend you do not do is simply screen all of these, look at the ones with the highest editing efficiency, choose those, and move on. And here's why. Often in the literature, you'll see people report editing efficiency. We're certainly guilty of it as well. Simply looking at sites where you change the DNA from the starting sequence. Now, if you think about it for just a moment, that's completely insufficient, right? If you're attempting to knock out a gene and your repair means that you actually just did a 3 nucleotide or 3 base, I should say, deletion at that site, the odds of actually achieving the functional outcome you desire are extremely slim. You probably just knocked 1 amino acid out of the open reading frame and otherwise are likely going to have a functional protein. So truly understanding the nature and the potential functional consequences of the changes you make at that site are incredibly important and have to go hand in hand with understanding the efficiency and therefore the potential potency of these particular reagents. And so as we have approached this problem, this is really why we rely heavily on our high throughput deep sequencing pipeline in order to truly understand what's happening. So I apologize, there's a lot on this slide and I'll walk you through the key details here. Fundamentally, after we've done our deep sequencing, at any individual target site, we want to understand the different collection of changes, DNA repair patterns, that occurred at any given target site. And we'll bin those. And so what you're looking at on the slide here in the main box, each horizontal bar is a different bin of sequences. So we're looking at a well that had thousands of cells in it. And so each bar represents many different cells that all experience the same edit, so the same cut and the same repair. And here we can see that little blue ball indicates wild type. So only 5% of the cells are unedited. Everything else is edited. And what's interesting is that there are actually only a small number of edits that make up the vast majority of changes that are observed in these cells. So for example, the most common edit, which is about 12% of this particular cellular population, is just a 1 nucleotide insertion. So those little blue boxes indicate an insertion, whereas the rest are deletions of various specific lengths at specific locations. So we've now gone on, obviously, and looked at many, many, many sites and have learned some very interesting things about DNA repair biology that really help inform the process of target identification. First and foremost, it turns out that DNA repair patterns are highly reproducible. And I'll show you some evidence of this. And that actually makes it much, much easier as you think about the experimentation necessary to identify a relevant guide for your target. So first, DNA repair patterns are reproducible across experimental replicates. So for example, we're looking at a particular guide RNA that targets the JAX1 gene, And when we run 3 experiments side by side by side, we get the exact same pattern of repairs. And so I'm showing you a slightly different format for digesting this information. It's a heat map that shows very much the same information as the last slide did, but in a way that we can start to easily compare across replicates or across experiments. So what you're looking at the very top is a map of the efficiency. And so you can see that the editing efficiency here is nearly 100% at each of these 3 replicates. The green box below that shows whether there have been any insertions. And if there's a 1 nucleotide insertion, we even bin it out by the specific nature of the nucleotide. We typically see that if there's a 1 nucleotide insertion, it's only one of the 4 possible bases that's the dominant insertion profile. And below that in blue is a heat map of the various deletions. And so you can digest this information quickly by just sort of scanning left to right. If the patterns look the same, it means that the repair outcomes were quite similar across these 3 experiments. So this was really remarkable to us to identify, and certainly outside of the standard understanding of DNA repair biology. There's a sort of a feeling in the field that that non-homologous end joining, NHEJ, this repair pathway, is very random, it's messy, it's noisy. It is messy, and that's helpful for gene editing, but it turns out it's not random, and that's also very helpful. We've also been able to demonstrate that this is true not only within one particular type of cell target, but across many different cells coming from the same species. And so I'm showing you here multiple replicates across multiple different cell lines now, again with the same guide RNA, where we're consistently able to achieve the same repair outcomes. Additionally, this holds true across different reagent delivery methods. So as you start to think both about the easiest ways to screen high throughput in your laboratory, there may be advantages for using methodologies other than direct delivery of the RNP. Each of those methodologies will still give you the same understanding and outcome of how these particular reagents will perform in a target cell. The timeline of those experiments may vary, but the actual DNA repair outcome will be the same. And so you can see here again, even when we switch to lentiviral delivery, where it takes days to weeks to achieve the same high efficiency editing, the DNA repair outcomes, yet again, are identical. And most importantly, this translates all the way through to human primary cells. And so now looking at a couple of different target sites in human T cells, looking at both TRAC, the TCR alpha chain, as well as beta-2M, which is part of HLA. In both of these cases, we're looking at triplicate experiments, each performed in cells derived from 4 different human donors. So looking at the ex vivo cell therapy literature, there's often conflicting information about performance of cells derived from different donors. Now in this case, we're lucky in that we're working with healthy cells from healthy donors. These are not cancer patients or cells that otherwise have been through recent traumatic experiences such as chemotherapy, but it's very promising to us to see that not only are these patterns conserved within, say, favorite cell lines in the laboratory, they're conserved across multiple humans as well. And I hope also with this slide I convince you not all guide RNAs result in the same pattern as well. Some are heavily biased towards particular deletions, others are heavily biased towards particular insertions. And so by having these incredibly rich data sets, we can now start to use bioinformatics to predict the potential functional consequences of this. And this allows us to integrate this information very quickly as we think about the best guides to target particular sites in the cell. Now, what's particularly remarkable about how the DNA repair works is that your target site choice is incredibly, incredibly important. And so what I'm showing you here are 3 side-by-side-by-side target sites where we've taken advantage of a particular location in the genome that has a run of 4 Gs. So the PAM, this motif, is NGG. So you can identify 3 potential target sites that overlap with offsets of just 1 nucleotide each. And what's really remarkable is with each of these 3, we see dramatic differences in the DNA repair outcomes. One of them is biased for a T insertion, one for a C insertion, and the other for small deletions. And so again, thinking about picking the right guide RNA for a particular target and for a particular functional outcome, this is one of the very strong reasons why we believe simply empirically testing each potential guide is by far the best way to get to the best answer of which guide will be best for your particular application. And what has been a bit of a head scratcher for us, but really interesting biology as well, is it's not just about the exact cut site even. So on the left-hand side of the slide, we're looking at two different guides that target on opposite strands aligned such that they actually result in a double-strand break at the exact same location in the genome. And yet, if you look at panel 1 versus panel 2, the DNA repair outcomes are completely different. Additionally, on the right, we're looking at two guides that target the exact same 20 nucleotide sequence, again from the opposite two strands, and again we see dramatically different DNA repair profiles. In data that I don't have time to fully walk you through today, we've published some and I'd be happy to answer questions later on. Really, what we think we understand at this point is that the exact 20 nucleotide sequence that's targeted by the guide RNA is the dominant feature that controls the DNA repair outcome. And so even though we think about maybe two target sites that are offset by a single nucleotide as being highly similar, apparently the DNA repair machinery sees these as fundamentally different. And that's ultimately what results in substantially different DNA repair outcomes. And so finally, the way we might integrate this entire workflow is to look at a target gene. For example, in this particular case, we're looking at TRAC, and simply design every potential guide RNA that could target a region within an exon in this gene. We'll create RNPs for each and every one of these, deliver them individually, side by side, in maybe 96 or 384 well plates, and ultimately go through the workflow that I described earlier. Now, in this particular case, TRAC is part of a cell surface receptor, of course, the T cell receptor, and so not only can we look at genotypic information through targeted deep sequencing, we can also look at functional outcomes and expression or loss of expression of the TCR on the cell surface. And so in this particular case, we're able to see, actually, I would say a pretty standard distribution, some guides that are really excellent, some guides that are pretty terrible, and a bunch in the middle. But what's also really satisfying is that there's a very good correlation in this case between high efficiency editors and high efficiency loss of TCR expression, which is what we're aiming to achieve in this. I'll say that's not always what we see. Sometimes we'll see incredibly high efficiency editing, but the actual nature of those changes does not result in high efficiency functional outcomes. And so that's why really combining all of this information is incredibly important. Now, given that we have just a couple extra minutes this morning, I'll take just a moment to talk about the next step in this potential process. And so as we might highlight, a few of these guides that I've circled in green here as the winners from this initial screen, the next important question to answer is how specific these reagents are. And so there's a methodology that we've developed at Caribou called CiteSeq in collaboration with DuPont Pioneer. That's a way to detect all of the potential off-target sites for a given guide RNA, and then ultimately measure whether off-target editing occurred at those sites or not. So the highlight of the methodology is to actually leave the cellular environment and go to a purely biochemical experiment. We'll isolate genomic DNA from our favorite cells of interest and digest it biochemically with RNPs. And we'll do a series of experiments of increasing concentrations. And then we have a slightly clever deep sequencing method where we can ultimately sequence everything in the reaction and specifically identify every site that was cut by that Cas9 RNP in vitro. That laundry list basically gives us the menu of potential off-target sites in the cell. We're then able to use that menu, go into our cells, maybe in this case primary T cells, and amplify and deep sequence each and every one of those sites to determine whether we see any off-target editing or not. Depending on the amount of effort that you've put into selection of your guide in the first place, you're typically able to find guides that have little to no evidence at the limit of detection of sequencing of any off-target effects in the cell. Of course, this audience knows better than most that simply making changes at the genetic level may not be all that matters. Understanding the functional consequences of any of those potential off-target effects is incredibly important as well. And so with that, I will wrap up here, say thank you to many of my colleagues who did this work, and would be happy to chat with any of you later. Thank you.